Hello and welcome to another Momento Malum today. In this episode, I'll talk about two news stories and the differences between quantitative and qualitative disease resistance traits. Our first news story is JBS opens a cleaning unit. JBS USA is one of the biggest US meat packers. Normally, meat packers use private sanitation firms to clean up the mess after the meat packing is done. One of the most predominant meat packing sanitation companies are Packer Sanitation Services Inc., or PSSI. This firm is an international organization that cleans slaughterhouses. However, not all is as it seems. Last February, it turned out that PSSI paid $1.5 million in penalties for employing the use of underage teenagers as cleaners for the slaughterhouse sanitation system. The age of these individuals have not been revealed, likely due to child safety laws. But ultimately, child labor laws were violated, and PSSI is now in hot water. JBS has created a company, JBS Sanitation, to do in-house cleaning for their slaughterhouses. The rival company of JBS, Cargill, is also moving away from PSSI. For our second and final news story, CNH, or Case New Holland, has increased the price of their vehicles. Due to the heightened demand for field crops in recent years, it's expected that there will be an increase in 16% sales and a 21% revenue increase. The strategy has already increased revenue by 29% in the first quarter of this year. Now on to the topic of plant. Next we'll cover the topic of the differences between quantitative and qualitative disease resistance. This builds upon a previous video, but we'll be covering the same material, albeit in more detail. Quantitative disease resistance is the most common form of disease resistance found within plants. It is controlled by many genes. And the full methodology of how it works is not fully understood. These traits are very environmentally dependent. In essence, a poor environment will reduce the effectiveness of this trait. Hundreds of specific responses are occurring at once to create quantitative disease resistance. This includes detoxifying toxins, cell wall thickening to limit the movement of pathogenic organisms, the release of inhibitor molecules, and pathogenic toxic materials such as proteins like chitinases and glucanases, as well as the release of free radicals to damage the pathogenic materials. This form of resistance is incomplete. However, it's not race-specific and works on all types of fungi and bacteria. But it's just like an immune system. In poor conditions, the immune system will work poorer than in better conditions. And if the conditions are really poor, the immune system will not be able to function properly, and the pathogen will be able to run roughshod over the plant, or the person in the case of the immune system. Since the resistance is incomplete, the pathogen is still able to reproduce, and in some cases succeed. This prevents the resistance from being overcome, because there's very little selective pressure for the pathogen to change. A robust quantitative resistance is controlled by a myriad of genetic changes, hundreds of changes if you will, and it'll take hundreds of mutations on the pathogen end to fully overcome it. All these factors limit the spread of resistant strains to quantitative traits, since they rarely ever show up. The problem is, of course, it's incomplete, and sometimes it will fail. And since the resistance is not complete, it has to be combined properly with management to limit the growth of the disease. This means fungicides if you're a conventional farmer, or organic techniques if you are an organic farmer. But in both instances, next we'll talk about qualitative resistance. But before we do that, we have to talk about the two different predominant types of disease out there: biotrophic and heterotrophic. Biotrophic diseases are pathogenic species that require living tissue to survive and function. If the colonized tissue dies, so do they, so they have to keep it alive, but still drain the resources. Examples include rust and powdery mildew. Necrotrophic species can survive on dead tissue, so it has no qualms about killing tissue or even the entire plant. This is because it can happily digest dead tissue as it would live tissue, so they go all out and drain the resources of the plant, and once the plant's spent and dead, it eats what's left. Fusarium and white mold are examples of such pathogens. The biotrophs function under a biochemical effector system where they use that effector to initiate the infection, allowing the species to infect their hosts and proliferate. The R gene codes for a protein that triggers rapid cell death when those cells come in contact, when the plant cells come in contact with a pathogenic protein. With the plant tissue dead, the biotrophic fungi cannot survive, and so it dies as well, and the rest of the plant continues to grow in peace. Eventually the pathogen forms a new effector protein through a single mutation, and those plants that have a mutation that have correspondence with the mutated effector protein will be able to survive, while those that had the previous adaptation or the previous R gene will die. In a conventional farm situation, R genes work fine for annual crops, for a simple reason that, at least within the framework of conventional farming, this year's variety of corn, soybean, or wheat will be replaced by a new variety next year. As such, one can cycle through these genes quite rapidly. On average, it takes about 9 years for such a trait to be overcome, making it ill-suited for perennial plants, especially trees, which are often grown for decades. The second weakness that this type of resistance system has is it does not work on necrotrophs, since it can eat dead material. So for tree fruit, and perennial crops in general, the ideal would be to focus on predominantly quantitative disease resistance, and this applies to both conventional and organic farming systems. And that about covers everything, but before we go, the sponsor's me. I have a Kijiji tutor ad. If you're interested in agricultural tutoring, the link is in the description below. And that about covers everything. Thank you for watching.